Hello again, Nicole Mashburn, and I'm going to talk to you a little bit about protein synthesis. So again, as we're getting into the tissues, we're going to talk about how things are made, and we're talking about cell division. And when you make more of anything, you've got to make more protein. So another part of this is going to be how to make our proteins. And again, this is something that if you've had biology, you know what I'm talking about. If you haven't, this is going to be new for you. So depending on your level of biology, this may or may not be review. So if it is review, great. If it's not, you can always go in your textbook and learn a little bit more about it. And I've got some review activities for you as well. So what is protein synthesis? Well, to synthesize means to make. So how do we make proteins? So first, let's see if you remember, where do we store the information on the blueprint for our proteins? In the DNA, in the nucleus. So the nucleus contains DNA, and DNA is coding for proteins. So what we want to do is we want to figure out a way to take that code, that blueprint that's in the DNA, and turn that into a protein. So do you remember what a protein is? A protein is a polypeptide chain of amino acids. That means you've taken a bunch of amino acids and linked them together to make a protein, okay? That's what we're talking about. So protein synthesis, remember, DNA is the blueprint for protein synthesis. So each gene is just a segment of DNA that codes for one protein or one polypeptide. So when someone talks about the genes that you have in your body, not your blue genes, but the genes in your DNA, each gene codes for a different protein, okay? And these genes, there's a code. Have you ever heard of the genetic code? Well, the genetic code is basically a triplet of nucleotide bases. So if you look at DNA, if you think back to our cell division, we talked about DNA replication, and you had those bases, and if you remember, I had the A and the T match up and the C and the G match up. Well, each one of those three sets of matches stands for a codon, and that is the code that we use for genetic information, and I'll show you that in a little more detail in just a minute. So each one of these triplets codes for amino acid. So you have three, this for this amino acid, then this codes for this amino acid, and this one for this amino acid. Again, that'll make a little more sense when you... Okay, so with protein synthesis, we've got to go through several steps. We've got transcription and translations where we're going to start. Transcription occurs in the nucleus and translation occurs in the cytoplasm. Now, what is transcription? Well, you have to make a copy of the DNA that then you can then use as your template to make the protein. And so I'm going to kind of explain why you have to do this. So take, for example, you have your notes and you make a copy of your notes for a friend. And then your friend makes a copy of that copy and gives it to her friend. And then that friend makes a copy of that copy of that copy and gives it to his friend. And you realize after a while, each copy gets worse and worse and worse. Well, if you want to make proteins, and you're going to be making proteins all your life, you don't want to make proteins from really bad copies. Okay? So every time you make a protein, you want to make a brand new copy, and that's what you want to make the protein from. Okay? Does that make sense? I think. So transcription is to make that good copy that you can then make your protein from. So transcription is taking your DNA and making a copy. And this is called your messenger RNA. And so the copy that we make is actually in the form of RNA and not DNA. It's single-stranded. This messenger RNA, which is now the copy that you're going to use to make your protein, then leaves the nucleus and goes out into the cytoplasm where it's translated. So you're going to take this code and translate it into an actual protein, okay? And this occurs in the rough ER using your ribosomes. So translation is making the copy, an mRNA copy, that you can then use to translate into a protein, all right? And again, there's some reviews that you can see in your textbook for more information, and also in Blackboard I've got a review for you. All right, so this RNA I keep talking about, there's actually 3 RNAs that are involved in protein synthesis. The first one is that messenger RNA. It's that good copy. It carries the instructions for making the polypeptide from the DNA inside the nucleus out into the cytoplasm. It carries it to the ribosomes, to the rough ER. The ribosomal RNA, or rRNA, that's the actual thing associated with the ribosome that translates the message. So it does the job. So it's actually translating, it's got the message, the messenger RNA, and it's actually going to do the job to translate that into a protein. Well, while you're actually translating the protein, you need somebody to bring you those amino acids. That makes sense? So you're sitting here and you've got the template and you're like, okay, I need these amino acids. I need somebody to bring them to me so that I can hook them together. So the transfer RNAs, they bring the amino acids that you need to link to your chain. That's pretty simplistic. I hope that made sense. All right. Okay, so let's take a look at how transcription works. This is pretty complicated. I really don't want you to learn all the steps of transcription, but what we're doing is we're transferring the DNA into a complementary copy of mRNA. And so this requires an enzyme, that ASE, RNA polymerase, is what you use to make that copy. And so essentially, like I said, I really don't want you to get into the details of what's going on, but this enzyme will unwind the DNA, and then it will allow a copy to be made, almost like DNA replication, except you're making an RNA copy. So you wonder, what's the difference between a DNA replication copy and a transcription copy? Good question. Remember we had that A, T, C, and G, those bases? Well, your RNA uses A, it uses C, it uses G, but it replaces the T with a U. So as it's going through and matching the copy, if it has an A, it's going to put a U there instead of a T, and then everywhere it's got a C, it'll put a G. If it's got a G, it'll put a C. So again, instead of using the T, it uses a U. That's really the main difference, and it's single-stranded. So you're making a single-stranded copy of your DNA, and you're using a uracil. That's what U stands for. Sorry, I should have said that. The U stands for uracil. I lost my pen. Uracil instead of the T. Okay, so you've made that copy. Again, don't bog down in the details. Now, once you've made this copy of mRNA, you're going to then take it out into the cytoplasm. And this mRNA copy, it's got all those little bases on it. Remember, it's got the A's, the C's, the U's, and the G's. And every 3 of these bases stands for an amino acid. So this is called your genetic code. So you've got the mRNA, okay, and it's just a long chain of these A's, C's, G's, and U's. And when we go out into protein synthesis, where we have the translation, the translator can read each one of these sets of 3 as an amino acid, okay? And so a set of these bases is called a codon, and each codon is an amino acid. And so if you ever had high school biology, your teacher probably gave you the genetic key and then gave you a strand of mRNA and said, okay, I want you to translate this into a protein. So you would look at your strand and say, okay, I have an A and a C and a U together. That stands for threonine. I've got a C, C, C. That would be proline. And you would link together your protein on paper. I'm not going to make you do that. But again, you're more than welcome to learn as much as you want to about it, especially if you're ever thinking about going back for your four-year degree, your BSN. You'll have to have some basic biology, and you'll see this. And you may do it micro as well, microbiology. All right, so again, for RNA, we've made our template. So we've transcribed, we've got a copy of our DNA in the form of mRNA, and that is now going to leave the nuclear envelope and go out into the cytoplasm. This is our mRNA. Now we need to translate it. We need to convert that message by linking amino acids together to make a protein. So here we have our rRNA, our ribosome. And our ribosome can read that message. So it knows, it can say, okay, these 3, that's a codon. It stands for an amino acid. These 3 stand for amino acid. Well, it's sitting there and it needs somebody to bring that amino acid to it. Okay? So transferring our RNA is going to have the complementary bases, the opposite ones, and it will come in and link and basically add those proteins one at a time, or those amino acids one at a time, making your protein chain longer. Okay, so again, don't get too bogged down in the details right now, because again, for this level course, I don't really need you to understand all the details. Just understand the difference between transcription and translation and which RNAs are used for what. Okay, so you use your messenger RNA for transcription, That takes the message to the ribosome, where the ribosomal the ribosomal RNA will read that, and the transfer RNA will bring the amino acid to it, and then you link them together and get your protein. All right. Now, so translation occurs in the rough ER, okay, in the rough endoplasmic reticulum. So here's my ribosome, okay, here's my mRNA. So you can go through if you want to read the individual steps, but you can see as the ribosome moves down the messenger RNA, it makes a long chain polypeptide. And this is going to be released into this rough endoplasmic reticulum. Now, once the protein is complete, that protein is then going to be put in a vesicle to be transported to the Golgi apparatus. Okay, so what happens in the rough ER is you just make the protein, but it's not ready. It's not functional yet. If you think back to chemistry, we talked about how proteins can have primary or secondary or tertiary structure. So now we're going to take those proteins, we're going to take them to the Golgi apparatus, and then the Golgi apparatus is going to do what it needs to do. It's going to process those proteins until they're in the state that they need to be for us to use them in our bodies. Okay, so again, the takeaway from this video is, what is protein synthesis? What does it mean? It's how we make proteins. It's how we link amino acids together to form polypeptide chains. Where is that message? It's in the nucleus. It's part of the DNA. The DNA has our genes. Each gene codes for a protein. Okay? We take a copy of our gene using mRNA, we transcribe, make a copy. Then we take that copy, put it in the cytoplasm to the rough ER, where we translate it. We turn that message into an actual protein. We read the message, and then we bring those amino acids in one at a time, link them together, and make the protein. Okay, that was protein synthesis in a nutshell, and that's really all you need to know about that right now. Thank you again.